So we are happy to have Thibault today. We're going to discuss spatial statistics in bioimaging analysis. Thibault is at the Pasteur Institute and is now has his group working on these topics, and probably you will discuss a bit today of the application with this artificial deep learning that we have developed, or we will see where you're.
A little bit.
All right.
Yes.
Okay. Thanks, Thibault.
Okay, so good evening. So today I'll describe the tools, the general tools that you can apply in bioimage analysis that use spatial statistics, and why do you need this type of tools to understand biological processes. So as David mentioned, I'm group leader now at Institut Pasteur in Paris, and you can contact me at this email. So this is just the outline of this course. So 5 main topics. First, I will describe few examples why the spatial analysis is important and where it can be viewed in biological imaging in general. And then I will spend some time on algorithms and methods that can be developed to characterize clusters in images. Then I will enter into the statistics and describe statistical tests that we build to test whether or not the object, so the biological objects, are randomly distributed or not. And I describe the extension to coupling. So when you have multiple type of objects and you want to analyze the spatial relations between different objects, and I'll spend at the end a few times about multiplex imaging and the challenges we can have with this type of new imaging data. So my very first slide is to say that actually here I describe tools that we develop for spatial analysis in biological imaging, but this is really ubiquitous in the sense that when you have objects that can be craters on Mars, galaxy cluster, or even stars, or people in a crowd, or even this, which describes leukemia cases in some neighborhood. The tools that are developed can be applied to a lot of fields. And looking at the distribution of objects is really a general and important problem. So in the first example, as you may know, in biological imaging, many images are acquired with fluorescence imaging, and then you can view some objects, fluorescent objects in an image that appear at spots because they are too small. So due to the diffraction limit of the microscope, you will have a lot of fluorescent spots in the image. And a natural question is to test or to describe whether or not these spots are clustered or randomly distributed, or if there is a distance between these objects. And why do we want to know that? Because it can give information about for example, here's the organization of the membrane, if the molecules that you are looking at are clustered in some domain of the membrane. So this is one important example. The other one is, and this is why this type of methods become more and more popular. It's because in fluorescence imaging now, people have developed super resolution imaging where you can extract the position of individual molecules in 2D or 3D. And so here, objects, so here molecules, they really appear at points because they are described through their position in a 2D. For example, here this is some synaptic molecules in 2D images, or even in 3D, and here it's two color 3D images where each sphere is one molecule in space. And so again, the natural question is whether or not they are clustered, or if there is any relations between the colors. And lastly, but and it's also maybe I don't know, but maybe most important application is within tissue, because now people are developing a new type of imaging in digital pathology, where you can acquire with quite high resolution an entire tissue in human through, for example, a biopsy. You can identify many types of cells within the tissue. And here again, so here this is an example where you look at the distribution of some immune cells in cyan in cancer. So here in orange, this is cancer epithelium. And on the right, this is different type of microglia in a human brain. And a natural question is to extract some information. So for example, here it's to describe is there any distance, or can we infer some processes about the immune response in cancer? And on the right, you can have this type of question, or whether or not the different types are close together, or close, or are they clustered in some regions of the human brain? So microglia, this is the resident macrophages of the brain. And so on the left, the different colors, this is obtained with immunocytochemistry. So this is just chemistry where you can use different color for different cells. And on the right, this is false coloring. Actually, there is a step of a deep learning classification of the cell, and then you have here up to 6 types of cells in the tissue. Okay. And yeah, I mentioned this before, but why do you want to look at this? So for example, here, this is the organization of cell membrane, the coordination of immune response. Here, this is a paper that we did for looking at the composition of synaptic vesicles. And here, this is a microglia, how they colonize the different brain regions. So this was just a brief and a general introduction about what type of data we are dealing with, and what type of questions we or others have in biological imaging. And now the second part is a little bit more technical. This is a brief overview of quite simple algorithms that can be used to characterize the object clusters. And before that, if you want to look at clusters from a mathematical point of view, you can ask different questions, which can be the cluster localization, where do you have clusters? There is some statistical question, the probability of real clusters versus clusters that chance when molecules or objects are randomly distributed. I mean, here, is this a real cluster or not? And here you have some clusters. So it's hard. I mean, this is a difficult question to know whether or not you have a real cluster or just a cluster by chance for just randomly distributed objects. And a last question is also, you want to quantitatively describe your clusters, the size, the composition, number of molecules per clusters, etc. So these are the 3 main questions I'm going to address in this part. So for the cluster localization, you need some method like local clustering algorithm. So you have your data set and you want to segment, design, find some cluster around with this type of algorithm. The second one, I will introduce a little bit more of statistics, and I will show that you can obtain this by building a test statistics and testing this under a null hypothesis, which is complete spatial randomness. And the last one is usually obtained by fitting a parametric model of clustering. I mean, there are other methods, but this is the 3 main methods I'm going to present you now. So for the local clustering algorithm, so a very naive almost everybody knows is like the K-mean algorithm. So you have a set of data and a set of observation, so endpoints, and you want to say, okay, this points belongs to n k clusters. So the idea is going to you are going to position position some cluster center at a mu, and you are going to how to say segment and put the different points in the different clusters so that you want to minimize the intra-cluster distance, and you want to minimize the you want to maximize the inter-distance between clusters and minimize the intra-distance between clusters. And so mu, this is the center of, so here, this is typically the red cross, because you need some iterative algorithm to position the red cross and then say that all the small red belongs to this red cross because they are the closest to the red one. This is a green and blue one. And typically, you have many K-mean clustering algorithms, but usually the idea is to position some initial position for your center of clusters, and then you're going to iteratively move so that you want to minimize the distance inside the cluster to the center and maximize the overall distance between clusters. So this type of method works pretty well, and you can find a lot of methods to partition your data in different clusters, but actually it does not really answer our problem, because first you need to specify how many clusters you want. Second, actually when you apply this type of methods, you realize that you really need a good initialization, and actually usually you are running this type of algorithm like 50 times and take the mean position for cluster centers. But most importantly, actually here I'm describing an algorithm that is partitioning, that is giving you a partition of your data and not really a clustering. I mean, even for an homogeneous distribution of points, I can still partition in 2, 3, 4 classes for clusters, but actually I'm just putting the different points in different class without really detecting a real clusters. So really an important algorithm to know to obtain clusters in a data set, and the data set can be points in space, but also any data set where you have a distance. And this paper is really important. It's like cited, I don't know, tens of thousands times. It's a DB-scan algorithm for density-based spatial clustering in application with noise. So it's quite simple. You need 2 parameters, the minimum number of points in around 4 points, and a search distance. So you specify a search distance epsilon, and then for each point of your data set, you look at the distance around, and if these points have more than here 4, but you can specify these parameters 3, 5. So here, if you have more than 4, 1, 2, 3, 4, then this point is a core point, and you do this for all the points. So you have all the red core points. Then all the points that can be that are at a distance less than epsilon from core points, that also belong to the cluster. So here I have my cluster in red and yellow. And all the points that are not at a distance less than epsilon from a core point, they are classified at R-plane. So very simple, but very efficient, because you have only 2 parameters. So the advantages are here. So there is no need to specify the number of clusters. This is automatic. And it can find arbitrary shaped cluster, because I have not described it, but you have a lot of clustering algorithms that are using Gaussian fitting. So you try to find Gaussian with some covariance in your dataset. Here you can find any type of connected clusters. And then it handles noise outlier quite efficiently and require only 2 parameters. But still some disadvantages because clearly going to depend on epsilon and the number of points you want to have in your core clusters. And so there is a question of calibrating these 2 parameters. And it can struggle in dataset with varying density. For example, if you have a big dataset and you have some areas with very few points, areas with dense points, and you have only 2 global parameters, epsilon and mean points, then you cannot handle your whole dataset. Then you have to refine the method by, for example, defining local parameters for epsilon and the mean number of points. And so, yeah, choosing an appropriate epsilon can be difficult. But still, I think this is one of the mostly used clustering algorithms. And here, this is an example that where we applied DeepCellMap with David, actually, on this dataset of Microglia cell. And so we can, so here, this is a cluster around, I think there's a blue here. And so you can define clusters and identify cluster of cells inside the tissue. And here's the polygon around, and this is just the convex pool of the cluster. Another method to find clusters, this is a Voronoi diagram algorithm. So here the idea is that you have a set, a domain, and you want to segment the domain in RK region, such that for each region RK, here all the pixels of the image are the closest to this point and the closest to this point. And the same idea, if you have 3, you are going to partition your domain in 3, 4 regions, depending on the number of points. And here, this is typically an algorithm that can be used for that, using seeds, and then you expand like a level set around your points, and you stop at the different boundary, and you get an efficient partition of your domain. So here again, this is a partition. So where are you going from? A partition to clusters. So here actually what happens is that when you have, so this is an example I took from a paper on single molecule localization microscopy. If you have a quite, I don't know what is red. I think the red is, okay. So if you have quite homogeneous distribution for the density of points in your image, then you will have a quite regular Voronoi diagram for your domain. If the distribution of density is like skewed like this, so you have a lot of area where the density is small, but then you have some regions where with a very high density of points. When you're going to do the Voronoi diagram, you are going to have smaller cells where the points are density are clustered, and larger cell around. And then you can identify clusters are adjacent small Voronoi cells. So here there is only one parameter. You have to define what is a small Voronoi cell, and you can also define some clusters with this method. Yeah. So I have described few algorithms to, visualize locally some clusters, but how can you be sure that you are looking at real clusters? Or because you can always take for example, for the DB-scan, you can always take an epsilon or a mean number of points that will give you some clusters. So in the lab, we built a test for this randomness, and a few other tests exist. But so I'm going to describe you how you can build a statistical test of randomness based on your point distribution. Okay. So here, but I will come to this after that. You have a null hypothesis, which is just a random Poisson process where you're going to put points around, and typically the mean number of points in a region, like for example, the probability to find one point in a region is proportional to the area of A. Very simple definition. And you want to reject this null hypothesis of spatial randomness, and so assess the presence of cluster in your point distribution. So if you want to do that, you need to choose a test statistics. So it can be a first order statistics based on local density. So for example, if you use a DeepCellMap algorithm with some parameter mean, points, and epsilon, you can look at the distribution of clusters areas that you have with your DeepCellMap, typically through a convex pool. And if you can define what is the distribution of clusters area under the null hypothesis, you can test whether or not you have real clusters in your distribution. But again, at each time, the difficulty is to define a good test statistics that can be characterized, defined, computed for a random distribution of objects. Also, for the Voronoi diagram, you can look at the distribution of cells area. So you do your diagram, you look at the size of the obtained cells and whether or not this size differ significantly from the distribution you would have for a random distribution of points. But so for these two algorithms, I don't know any characterization for this distribution. I mean, I'm not sure it has been computed yet. So usually for testing the spatial randomness, people use second order statistics. So second order because you look at the distance between neighbors. And the two main statistics used here are typically the distance to the nearest neighbor. So in the plane, in the space, the distance to the nearest neighbors for a random distribution has been defined and characterized. Or the other example, mostly used, is replace K functions that I'm going to introduce. And for these two, you can compute the distribution of these two functions when objects are randomly distributed. So for the replace K function, this is the one that we have been working on in the lab. You give a parameter R, which is a search distance parameter that can vary. And around this, you're going to look at the number of, so for each point X in your distribution, you're going to count the number, how many points Y you have at a distance less than R. Here, this is a normalization factor. So this is the whole size of the domain divided by ln minus 1, where n is the number of points. And here, this is a boundary correction, because actually it's quite important. So here, this is a type of boundary correction. Why do you need a boundary correction? Because typically for all the points that are close to the boundary, if you look at the distance r around, you see that naturally you will have, for a random distribution, you will have less neighbors for comparing with a point that is right in the middle. And typically what happens is that if you have a random distribution and if you take a point right on the boundary, you expect almost half, neighbors compared to points that are at the center of the domain. And Ruchiy is an English statistician that worked on this in the 70s. Yes, this is a book that has been published in the 80s. And he recommends to use this type of boundary correction because it looks a bit weird, but it's good for computing some statistics on the replace function. So the idea is for this boundary correction, so you have a first point x, a second point y, and your parameter r. And so if you look at the circle DB, so this is a circle that is centered on x with radius x minus y. So typically here you have this x, and so for example, let's say that this y, there is a small shift, but this is a circle that is centered on x passing through y. Then you look, so the total, so this is a total perimeter, so this is 2 pi dot distance, and you divide this by the proportion of the circle that is inside the domain. And so typically, if x is exactly at the boundary, then x here, this is 2 pi times distance, and here this is 2 pi times distance, but divided by 2, because you have only half of the circle that is inside the domain. And then the boundary correction is 1 divided, it's like 2 pi divided by pi, so it's 2. So here we are going to count twice the point y to compensate for the fact that the point x is right on the boundary. It's a little bit technical, but this is just to let you know that this difficulty arises when you look at the distribution of points in a closed domain, and it's quite important. And actually, this is what complicates all the computations, because when you look at books and you look at what people did for example, deriving the distribution of distance to nearest neighbor, all the time this is performed in space with some density of points rho. But in real life and in biology, it never happens. You never have an infinite space plane. So all the time you have some boundary you have to deal with. And a very common hypothesis in spatial statistics is the complete spatial randomness. So I'll talk a bit about this. So it's really a point process where events occur randomly, completely randomly, also known as homogeneous spatial Poisson process. And there is only one parameter, rho, the density of points. So typically, a good estimate of rho for a real image is the number of points divided by the size, the area of the domain. And so there is multiple definition. One is that the probability to find exactly, k points within the area V is given by a Poisson distribution. So V rho, so this is rho density, V size of V, to the power of k. So this is a Poisson distribution, so the probability of finding k points for a process with density rho in a domain, in an area V. So as I mentioned before, rho can be quite naturally estimated as the total number of points you have divided by the size of the domain. And as I mentioned before, another view of the main hypothesis is to say that the mean number of points in V, inside V, is rho V, which is n V divided by omega. So if you remember before, I mentioned that in an homogeneous Poisson process, the probability to find a given point in V will be the size of the V of V divided by omega. So it's quite a natural definition. Okay, and so here this is typically the computations that have been performed by Ripley in the 80s. So he computed the mean of the k function R for spatial randomness, and so you expect, so you have some normalizing factor, but at the end, the mean number, or the mean of kr is the size of the size of the search domain. PR square, and the variance. So here the computation is it's a bit tricky, but if you assume that the boundary is locally straight, so you can do some computation, then and you can read the book, but he derives the variance, which is this term. So with beta PR square divided by omega and gamma, this is the perimeter, the domain, R divided by the domain. And here you have strange constants that are coming from numerical approximation of some integrals. So you have some integrals that are, and you can get this numerical approximation. And that's pretty great because you know now, so for K, and you see I mean, if you didn't have this boundary correction, this expression would be much shorter. So I think typically you would have no gamma. So all these terms would disappear and you would have a very simple term. But because of the boundary correction, you obtain this type of numerical integration, which are not easy to anticipate. And so this is a type of statistics you can use because now, so let's introduce the normalized. So you take your function k, you take minus mean and divide it by square root of variance. And so this is a normalized random function because the mean of k cleared is 0 and the variance is 1 now. And you can define the quantile. So for this, you need to know what is the distribution of k. And actually, what people have shown is that when you have a sufficient number of points, the k-tile, type k, is a normal distribution. So the quantiles are just the quantiles of a normal distribution, meaning that the probability that normal distribution is above the alpha is equal to alpha. So typically, if you want what is the probability to be at 85%, the z is at 1.64. So this is completely tabulated. And actually, we work a bit on this because usually in many applications, what happens is that the number of points is not that big. And what happened, because typically if you look at the mean number of neighbors, which is a kn divided by omega all around, the mean number of neighbors at distance r, when you don't have so many, not so many points, the problem is this mean number will be always positive. But so if you shift this distribution to the left, it cannot be negative. So what happened in this type of problems is that this distribution, this Gaussian distribution, which is centered, you have the variance and it's centered. When you are going to shift this, because the number of points is not that large, then what happens is that you're going to have a distribution with a skewness that is positive. So it's because you have more positive events than negative because it cannot be less than 0. And a kurtosis that is above 3. This is because the distribution is going to be larger. So kurtosis is more or less the kurtosis. This is the size of the tail, if this is a heavy tail or not, and the skew is the asymmetry of the distribution. Okay, so this is a bit technical, but I wanted to mention this because in some application, you have to be careful that if you have not so many points, the distribution is not Gaussian, because the Gaussianity is the law of large numbers. So if you have not so many points, the distribution is not Gaussian. And this is typically what happens in different applications, because you are looking at a variable that needs to be positive. And to be positive, you need to introduce a skewness and a kurtosis. but it's not, it's okay, it can happen, and you can still correct for this with the Cornish-Fisher expansion of quantile. It's not very well known, but this is a very nice way to correct for the quantile based on this skewness and kurtosis that you can observe in many distributions. So the quantile is actually approximately Z alpha, which is the quantile of normal distribution. Gaussian approximation is here. And then you have this term. So let's just decompose it a bit. So typically for a normal distribution, the skewness is 0. So all these terms disappear, and the kurtosis tends to be equal to 3. So this term disappear, and here you have a skew squared. So all these terms disappear. So all these terms are 0 for Gaussian distribution. But when the skew is positive and when the kurtosis is above 3, you have this new term, and it gives you a very good approximation of your quantile. Yet, you need to compute the skewness and the kurtosis. So it can be done. I mean, I did it, but I won't give you the whole formula. It's quite big, but still you can. This is something that you need to keep in mind, that sometimes life is not Gaussian, but you can have some approximation, interesting approximation. And this type of approximation is largely used in finance modeling, because typically, for example, if you look at a value of stock, I don't know, something that cannot be too positive, usually you have a kurtosis that is a heavy tail distribution, because you can have large positive value, not so negative, and the skewness of the distribution. And the last part for this clustering is the quantification of cluster. And so here I'm dedicated. So you can, for example, if you use density-based clustering or, I mean, a local method for clusters, you can still extract your clusters and measure their size or their composition. Here, I just wanted to mention this. If you have a K distribution like this, that is above the quantile, so here you have something, and based on the maximum and where it is reached, if you have a model for the clusters, you can extract the mean size of clusters, which can be interesting. So this is just to illustrate that you can also have access to some characteristics of the cluster based on fitting a model to a curve. And so here I chose quite general matter and cluster process. So in this model, you have clusters with radius R, RC, and you are going to position these clusters randomly in the domain. So the cluster centers, this is a Poisson process with some intensity lambda c. So they are randomly distributed. So if lambda c is constant, and lambda c gives you the mean number of clusters. And in each cluster, you're going to put a proportion of the total proportion of points. So you have a 1 minus p that are randomly distributed, and a p points that are inside the clusters. And based on this, you can derive what is the expectancy of your K type given this proportion of cluster, lambda c, RC, and p. And actually, it's not that big, but you can show that this is 1 divided by n, and you have this lambda c, RC, and p. And so you can have this type of function. This function can be found in textbooks. And so if you derive this and take 0, you can show that the maximum is reached. So at R max, so the derivative, so dr is equal to 0. And if you invert this, a numerical inversion gives you that R max, so the radius is approximately R max divided by 1.3. This is just an example to show that you can, even if you have a global description of your point process, with some parametric modeling and fitting, you can have access to some characterization of the cluster. Okay, so this concludes the first part, a bit technical about clusters, how you cluster data, how you cluster spatial points. And all the second part is what happens if you have many different types, at least two different types of cells, objects, and you want to look at the spatial association between the two. Peut-être on peut faire une pause, ouais, 5 minutes.
Okay.
Okay. Yes, we can do a pause like 10 minutes and we come back at 6, 5.